Our next topic is shikimic acid pathway. So, coming to shikimic acid pathway, the name shikimic acid, it comes from the Japanese flower, that is shikimi. Okay. The shikimi, it is also known as Japanese star anise or star anise. The botanical name of this flower shikimi is Elysium anisatum. So from which it was first isolated, and this was isolated in 1885 by Johann Friedrich Ekman, and almost after 50 years the structure was elucidated. The structure of shikimic acid was elucidated. Okay. So generally it occurs in plants, fungi, bacteria, but the shikimic acid pathway does not occur in animals. Okay. So the main function, the main role of this shikimic acid pathway is it involves in the biosynthesis of aromatic amino acids such as phenylalanine, tyrosine and tryptophan. So if you see here, the phenylalanine and tryptophan, these two are essential amino acids. Okay, as the the shikimic acid pathway does not occur in the within the animals and human. So in human, so there is no production or synthesis of phenylalanine or tryptophan. So they are called as essential amino acids because essential amino acids means the amino acids which cannot be synthesized by the body, and it has to be provided from the external diet. Okay, so it is the metabolic pathway which involves in the biosynthesis of aromatic amino acids. They are also known as phenyl propane derivatives or C6C3 derivatives, and it involves in the synthesis of amino acids such as phenylalanine, tyrosine and tryptophan. Okay, so here the phenylalanine and tryptophan are the essential amino acids, and coming to the tyrosine, it is synthesized from the phenylalanine. So it is categorized under the non-essential amino acids. So when you supply or when you provide this phenylalanine from external diet, the tyrosine will be tyrosine. It utilizes the phenylalanine and synthesizes tyrosine within the body. Okay. So, as the shikimic acid pathway does not occur within the humans, so there is no production of phenylalanine, tyrosine and tryptophan. Okay. Next. So, it involves in the biosynthesis of C6C3 units. C6 means phenyl, C3 means propane. Okay. It involves in the biosynthesis of phenyl propane units. So from the intermediate, so coming to the precursors of this, generally the intermediates of glycolytic and pentose phosphate pathway involved in the shikimic acid pathway, that is involved in the synthesis of shikimic acid. The intermediates of glycolytic pathway, this is nothing but phosphoenol pyruvate. It is an intermediate of glycolysis. And erythrose 4-phosphate, it is an intermediate of pentose phosphate pathway. These two act as starting materials or precursors of the shikimic acid pathway. And the amino acids, it also contains nitrogen in its structure, right? So the amino acids, it contains a carboxy group and one amino group. So the nitrogen source, the nitrogen is obtained from other amino acids. Other amino acids are also participating. They are also involved in these reactions. Okay. So the other amino acids which contributes nitrogen are glutamate, glycine and serine. Okay. This is about the precursors of the shikimic acid. Shikimic acid pathway is a very important pathway. So this mainly involves in the biosynthesis of aromatic amino acids, mostly that is nothing but phenyl propane derivatives of C6C3 units. And coming to the precursors, it mainly involves the intermediates of glycolytic and pentose phosphate pathway. The intermediate of glycolysis pathway is phosphoenol pyruvate, and erythrose 4-phosphate. It is an intermediate of pentose phosphate pathway. So nitrogen, it is obtained from the amino acids such as glutamate, glycine and serine. So moving on to the schematic representation of Shikimic acid pathway. So carbohydrate metabolism, obviously it involves in the Shikimic acid pathway. So from glycolysis we are getting intermediates, and also from pentose phosphate pathway also we are getting the intermediates which involve in this Shikimic acid pathway. Okay, so they undergoes various reactions, and after it forms intermediates and then forms the Shikimic acid, and lastly it comes to coricimate. Here in the Shikimic acid pathway, the coricimate is important because from the coricimate you get the phenylalanine, tryptophan and tyrosine. Okay, so these are the aromatic amino acids. Again from these amino acids we get the flavonoids, coumarins and lignins, that is from phenylalanine, and from tryptophan we get the alkaloids, and from tyrosine cyanogenic glucosides or glycosides and plastoquinone and tocopherol. Okay, and not only the amino acids from the coricimate, we also get the folate, ubiquinone. This is a schematic representation or overall representation of the Shikimic acid pathway. Okay, the metabolism of carbohydrates undergoes various reactions and finally forms the coricimate. From the coricimate we get the phenylalanine, tryptophan and tyrosine, and these amino acids further synthesizes flavonoids, coumarins, lignins, alkaloids, glycosides, etc. Next. So this is the role of Shikimic acid in a living organisms. Okay, so from Shikimic acid we get chlorogenic acid. Okay, so the precursors are erythrose 4-phosphate and pentose phosphoenol pyruvate and erythrose 4-phosphate. So Shikimic acid, so it is synthesized from the precursors are phosphoenol pyruvate and erythrose 4-phosphate. Okay, so from there we get Shikimic acid, and from Shikimic acid we get the chlorogenic acid. So Shikimic acid undergoes further reactions and forms the chlorosemic acid. Okay, so this chlorosemic acid directly gives rise to the phenylalanine. Here also some intermediate reactions are there. It undergoes reactions and then gives phenylalanine. From phenylalanine again we get tyrosine, and this chlorosemic acid, it forms anthranilic acid. From the anthranilic acid we get tryptophan. So this tryptophan it helps in the biosynthesis of indole glucosinolates and indole phytoalexins and tryptamine, indole and quinoline alkaloids, indole acetic acid. Okay. And phenylalanine, it mainly helps in the biosynthesis of phenylethylamine and cinnamic acid, and also isoquinoline and benzyl isoquinoline compounds. So cinnamic acid, further it gives flavonoids, anthocyanins, lignin precursors, coumarins and phytoalexins, salicylic acid. Okay, so this is how it produces various primary and secondary metabolites. And in the same way, from tyrosine it helps in the biosynthesis of morphine, betalains and cyanogenic glucosides and some other alkaloids. Okay, so this is the importance of Shikimic acid pathway. Okay, Shikimic acid pathway, it involves in the production of biosynthesis of large number of primary and secondary metabolites. So coming to the pathway, so this is the Shikimic acid pathway, right? So here we can see erythrose 4-phosphate and phosphoenol pyruvate. Okay, these two are the precursors. Erythrose 4-phosphate, it is obtained from the PPP, and phosphoenol pyruvate, it is obtained from the glycolysis. Okay. Pentose phosphate pathway and glycolysis. These two are the intermediates, right? So these two acts as the precursors in shikimic acid pathway. So here various steps are there, right? So here we can see the steps involved in the shikimic acid pathway. So this is the first step. Okay, in first step, the phosphoenol pyruvate and erythrose 4-phosphate, these two combine together and undergoes aldol condensation. Okay, so these two compounds, they undergoes aldol condensation and forms 3-deoxy D-arabinoheptalosinic acid 7-phosphate. This is also known as DAHP, 3-deoxy arabinoheptalosinic acid 7-phosphate. Okay, so here you can see. So here we have 4 carbons and here we have 3 carbons. So here we have 4 carbons, erythrose and phosphoenol pyruvate 3 carbons. So here this we are getting the DAHP. Okay, these two undergoing condensation, and so we are getting this. Okay, so this you can see here, it is joining here on CH2. Okay, so here we have, this is our phosphate group, erythrose phosphate. 1, 2, 3, 4, 1, 2 and 3. So, second step. So coming to the second step is cyclization of DAHP. So here there is no cyclic compound. Okay, so it is an open chain, right? So here we have the phosphate group and keto group. Okay, so now it is undergoing cyclization in second step. So the second step, it undergoes cyclization in presence of cofactor, that is NAD, nicotinamide adenine dinucleotide. Okay, and cobalt. These two acts as cofactors in this step, right? So in presence of NAD and CO, it undergoes cyclization, and this reaction is catalyzed by 3-dehydroquinic synthase. Okay, so after cyclization, it forms 3-dehydroquinic acid. So here you can see this is our C7 and this is our C2. So this keto group is converted to hydroxy group, okay, and the bond is formed here, and the phosphate group is removed. Okay, so the phosphate group is removed, and from here the hydroxy group is converted to keto group. Okay, one hydrogen from here and this phosphate group. Okay, dephosphorylation, and here hydrogen is added, so the keto group is converted to hydroxy group and cyclization takes place. Okay, the bond is formed between the C7 and C2. Okay, so and it forms 3-dehydroquinic acid. Okay, so next step. So in the third step, the 3-dehydroquinic acid, it undergoes dehydration and means removal of water from the compound, right? And this step is catalyzed by an enzyme, that is 3-dehydroquinic dehydrase. So here we can see (3-dehydroquinic acid). So this is our 3-dehydroquinic acid. So from here removal of water takes place. Okay. So here we have hydroxy group. This is our hydroxy group. One hydrogen from here, it will be eliminated in the form of water. So the hydrogen bond. So I'll show you. So now here, so here you can see this OH, this OH and this hydrogen. Okay, so from here one hydrogen is leaving. So here you can see this hydrogen hydroxy group, and hydrogen from here, okay, will be removed in the form of water. Okay. This hydrogen, so this bond shifts here. So here we got this double bond, right? See, uh, dehydration. So this OH group and H from this point, okay, it will be eliminated in the form of water, and the bond, it shifts here. You will get a double bond here, okay, and forms 3-dehydroshikimic acid. Okay, so next, uh, 3-dehydroquinic acid, it undergoes dehydration, and with the, that means removal of water, it forms 3-dehydroshikimic acid. So this reaction is catalyzed uh, by an enzyme, that is 3-dehydroquinic dehydratase. The next step, fourth step is the 3-dehydroshikimic acid undergoes hydration in presence of NADPH. Okay, so this NADPH, it gives an hydrogen molecule to the 3-dehydroshikimic acid and helps in the formation of shikimic acid, and this reaction is catalyzed in presence of shikimic acid dehydrase. So here we can see shikimic acid, okay, this time. So we have 3-dehydroshikimic acid, okay. if you see here we have a keto group at third position, right? So here what happens, in the fifth step, fourth step, the hydration takes place. Okay, nicotinamide adenine dinucleotide phosphate. It gives one hydrogen molecule and helps in the conversion of keto group to hydroxy group. Right? So here we can see, here we have a keto group. So here we have a keto group, right? So here we got a OH hydroxy group. Okay, so one hydrogen molecule is given by the NADPH. Okay, so this is the fourth step. Okay, in the third step, the 3-dehydrochromic acid is converted into 3-dehydroshikimic acid, and in the fourth step, the 3-dehydroshikimic acid, it undergoes hydration in the presence of NADPH and forms shikimic acid. So this shikimic acid is an intermediate which involves in the shikimic acid pathway. Okay, moving on to the next step, that is fifth step. So in the fifth step, the shikimic acid, it undergoes phosphorylation. Okay, this phosphorylation takes place in presence of ATP. Okay, so adenosine triphosphate. So here this triphosphate is converted to diphosphate. So here one phosphate molecule is taken by this shikimic acid and forms shikimic acid 3-phosphate. That is, in the fifth step, phosphorylation takes place. So shikimic acid is converted to shikimic acid 3-phosphate in presence of ATP. Okay, so ATP gives one phosphate molecule to the shikimic acid and itself gets converted to ADP, adenosine diphosphate. Okay, this reaction is catalyzed by shikimate kinase enzyme. Okay, so here we can see, so in this both the fifth and sixth steps are combined. So just see this part, only this part. Okay, so here we can see that the shikimic acid, here we have the hydroxy group. So here it is undergoing phosphorylation. Okay, so during the phosphorylation, what happens, the phosphate group comes and attached at this position. Okay, so here you can see the phosphorylation at this position, right? So it forms shikimic acid phosphate. So shikimic acid phosphate. Okay, from this side 1 to 3rd position, right? So next step is phosphoinhalation. Okay, phosphoenol pyruvate. So here we already have. So the phosphoenol pyruvate, again one more molecule comes and attached to this shikimic acid 3-phosphate. Okay, so addition of phosphoenol pyruvate followed by dehydration. Okay, this reaction is catalyzed by the enzyme EPSP. Okay, so EPSP means 5-enol pyruvyl shikimic acid 3-phosphate. That is nothing but EPSP. Okay, so in the sixth step, the shikimic acid 3-phosphate with the addition of phosphoenol pyruvate and elimination of water, with the loss of water molecule, it forms 5-enol pyruvyl shikimic acid 3-phosphate, that is EPSP. So here we can see this is EPSP, right? So both the steps are combined here, both 5 and 6 steps, phosphorylation and addition of phosphoenol pyruvate. Okay, so the enol pyruvate joins at this position. Okay, so now this structure is, the numbering is changed, 1 2 3 4 5. Okay, So the name of the compound is 3-enol pyruvyl shikimic acid 5-phosphate. Okay, so here we have the larger molecule, so numbering starts from here, 1 2 3. So 3-enol pyruvyl shikimic acid, and this is the 5th position, 5-phosphate. Okay, so this is about the 6th step. Next, coming to the 7th step. In the 7th step, the EPSP undergoes dephosphorylation. The phosphate group is removed from the EPSP. This reaction is catalyzed by the enzyme poricimate synthase. So here we can see, right? So here we have the phosphate group at 5th position and enol pyruvate at 3rd position. Okay, so here what happens in this step, in the 7th step, the phosphate group is removed. Okay, so here we have H2PO3. So from here one hydrogen molecule, so from here one hydrogen molecule involved in the reaction, and this bond shifts here, and this bond it shifts here. Okay, so here you can see there are 2 bonds formed, right? Okay, so hydrogen from here, and this bond is shifting here. Okay, the phosphate group is removed. So here we have 2 double bonds in the structure, right? So in the 7th step, the 3-enol pyruvyl shikimic acid 5-phosphate, it is undergoing dephosphorylation, that is the removal of phosphate ring. So here hydrogen from this carbon is involving, right? So the hydrogen and this phosphate group, so this bond shifts here and this bond shifts here and forms poricimic acid. This is our poricimic acid. Next, 8th step. In the 8th step, poricimic acid, it forms prefinic acid on one hand and anthranilic acid and on the other hand. From the anthranilic acid we get the tryptophan, and from prefinic acid we will get the phenylalanine and tyrosine. Okay, so first we will see this anthranilic acid. So poricimic acid, it forms anthranilic acid. So here we have anthranilic acid. This is anthranilic acid. This is our 8th step. So poricimic acid, it forms anthranilic acid in the presence of anthranilate synthase, and this further undergoes reactions, intermediates are formed, and finally it gives phosphoribosyl anthranilic acid. Okay, so this in presence of an amino acid, that is serine, and it forms tryptophan in the 9th step. Okay, so the reaction is catalyzed by tryptophan synthase enzyme. Okay, so here anthranilic acid and phosphoribosyl anthranilic acid. Okay. Finally, so these are the intermediates formed, and finally it forms tryptophan. Okay, so from poricimic acid we also get para amino benzoic acid, that is PABA. Okay, so that is different. Okay, so in the 8th step, the poricimic acid is converted to anthranilic acid in the presence of anthranilate synthase, and from sequence of reactions, the anthranilic acid, it is converted into phosphoribosyl anthranilic acid, and in presence of tryptophan synthase and serine amino acid, it forms the tryptophan. This is our 9th step. And coming to the 10th step, poricimic acid is converted to prophenic acid. Okay, and this prophenic acid undergoes oxidative decarboxylation. That means it undergoes decarboxylation, removal of CO2 and dehydration. Okay, the water molecule is also removed and forms phenyl pyruvic acid. So here we can see the carboxylic acid group is removed, CO2, and here we have a hydroxy group, so that is also removed. Okay, so directly from the corysinic acid it forms prophenic acid. So this prophenic acid, it undergoes decarboxylation and dehydration, right? So here CO2 is removed and from here H2O. Okay, so the bond shifts here and so the shuffling of the bonds is taking place and forms a phenyl pyruvic acid. Okay, so this is our 11th step. In the 11th step, prophenic acid undergoes decarboxylation and dehydration, forms phenyl pyruvic acid, and in the 12th step, phenylalanine transaminase and one amino acid, that is glutamate, it forms the phenylalanine. Okay. So in presence of phenylalanine, transaminase and glutamate amino acid, it forms the phenyl pyruvic acid, it forms the phenylalanine. Okay. So next step, 13th step. So in presence of in NAD, removal of decarboxylation takes place. So here we can see prophenic acid. So it is undergoing decarboxylation. The hydroxy group remains same. So only the CO2 is removed, right? And shuffling of bonds, right? So the hydroxy group is there. 4-hydroxy phenyl pyruvic acid is formed. Okay. So in the last step, that is 14th step, reductive transamination takes place in presence of tyrosine transaminase and glutamate. Okay. Here also the amino acid involved is glutamate, and the enzyme is enzyme which catalyzes this reaction is tyrosine transaminase. Okay. So this undergoes reductive transamination. Okay. So and forms tyrosine. So these are the 14 steps involved in the biosynthesis of shikimic acid pathway. So this is the same picture showing this shikimic acid pathway, phenol, phosphoenol pyruvic acid and erythrous 4-phosphate. In the first step, it is undergoing condensation and cyclization, right, and dehydration, and then it forms the shikimic acid in the fourth step, and from the shikimic acid it forms shikimic acid 3-phosphate, and again one more phosphoenol pyruvic acid is added and then forms the corysinic acid. From corysinic acid we get two compounds, that is anthranilic acid and prophenic acid. Anthranilic acid, after some intermediate reactions, it forms the tryptophan in presence of serine, and prophenic acid, it forms the phenyl pyruvic acid. It undergoes decarboxylation, right? And here dehydration and decarboxylation. Here it undergoes decarboxylation and forms para-hydroxy phenyl pyruvic acid. Okay, the phenyl pyruvic acid gives phenylalanine, and para-hydroxy pyruvic acid, phenyl pyruvic acid gives tyrosine. Okay, so this is about the shikimic acid part. So here also you can see. So here this is phosphoenol pyruvate and this is erythrous 4-phosphate. So here you can clearly see it is indicated with different colors. So here we have dihydrophenate here this is the second carbon, COOH and CO. Okay. So here the H groups uh, denoted with this D. Okay. So 3-dihydrophenene shikimate. So if we have H, that is shikimic acid. Right? So enol pyruvyl shikimate 3-phosphate, corismate, and from corismate rafinic acid, phenyl pyruvic acid, phenylalanine. And here we are getting tyrose. So from the corismate again we get the anthranilic acid, from which we will get tryptophan. So biosynthesis of alkaloids. So most of the alkaloids are derived from the amino acids such as precursors such as ornithine. So till here this is about the shikimic acid pathway, and this shikimic acid pathway, it is very important in the production of various secondary metabolites, right? So we also have alkaloids from different amino acids, not only from the aromatic amino acids. We also get most of the alkaloids from the amino acids like ornithine, arginine, lysine, apart from this phenyl, tyrosine and tryptophan. Okay. So this is about the shikimic acid pathway.